The path from research to the clinic is often impeded by the very materials designed to support progress. Off the shelf media and buffers may meet early research needs, but they frequently create challenges during scale up, tech transfer, and regulatory review. And customization, it can feel out of reach as it's often considered too complex, too risky, or too slow. But what if there was a way to simplify media development without compromising on consistency, compliance, or quality? I'm Miles Skoda. This is the CellAndGene.com virtual event series, and today we're talking Is Your Media Silently Holding You Back? The Case for Optimized and Excipient GMP Cell Culture Solutions. Now, joining me for this presentation is a returning guest and industry expert from Nucleus Biologics, Director of Research and Development, Dr. Asma Ayari, who is going to look at how QuickStart Media simplifies custom media development by providing pre optimized formulations that can be easily tailored to specific cell types and processes. She's going to cover the critical role of GMP-grade excipients in ensuring consistency, manufacturing efficiency, and regulatory compliance for cell culture media and buffer manufacturing, and show how to streamline the transition from RUO to excipient GMP manufacturing with a combined approach that reduces risk, accelerates timelines, and supports successful clinical translation. So we will be holding a Q&A session following this presentation. Please feel free to submit questions as we go along. We'll get to as many as possible during that Q&A, and any that we don't get to, I'll pass along to Asma and the team for potential follow-up after the event. And then just a bit of housekeeping. If you run into any issues today, please try refreshing your browser first. If your problem persists, you can submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often much less. You get a link to it, but you can also just use the same link that you used today. So with that said, let me turn it over to Asma to get started.
All right. Thank you, Miles, for the introduction, and certainly happy to be here today. My name, as Miles mentioned, Asma Ayari, head of R&D at Nucleus Biologics. So I mainly work on developing formulation for cell and gene therapy. So at Nucleus Biologics, our mission is to speed the time from scientific discovery to cure by driving reproducibility through precision cell culture products and tools. And transparency is at the heart of everything we do. And we deliver flexibility at every single level of your media development manufacturing. So whether it's specialized packaging configuration or meeting specific regulatory requirements, we tailor our products and services to fit your needs. As cell and gene therapy advanced toward commercialization, there's been a dramatic increase in demand for excipient-grade manufacturing standards. And beyond our existing ISO 9001 and ISO 13485 certification, we pursued and achieved both EXCiPACT certification and PAMS compliance. This ensures our manufacturing processes meet the rigorous standards required for excipient-level production, giving you confidence in quality and regulatory compliance. Scalability is another cornerstone of our approach. We understand that successful therapies don't begin at commercial scale, they start in the lab. You need a partner who can grow with you from early stage R&D through GMP manufacturing. That's why we've built a streamlined transition pathway that takes you from benchtop to bioreactor with comprehensive support for development, optimization, and validation at every stage. So right now, multiple companies rely on Nucleus Biologics for commercial scale production of clinical grade products. Our media and buffers are already in therapies reaching patients. So when you partner with us, you're working with a team that understands the journey from research to clinic and has the transparency, quality system, and proven track record to support you at every step on the way. Cell and gene therapies represent a transformative approach to treating a wide range of diseases, from rare genetic disorders to cancer. But unlike traditional pharmaceuticals, cell and gene therapies are living medicine that either modify the patient's own cells in autologous therapies or utilize donor or engineered cells in allogeneic therapies to restore and enhance biological function. So the complexity of cell and gene therapy manufacturing stems from its biological nature, requiring precise handling, manipulation, and expansion of living cells. So as you can see on this graph here, multiple moving parts are involved for a successful therapy, such as the cell source, the vector design, the manufacturing facility, the labware used, the protocols applied, the reagents composition, and source. All these parameters need to be considered and evaluated to allow for an efficient and successful development phase, especially that they affect each other. From cell collection to genetic modification to expansion, harvesting, and delivery, every step must be carefully controlled to maintain cell viability, function, and safety. And at the heart of this process are cell culture solutions, including media, supplements, and buffer assistance. In this presentation, I will be emphasizing on the impact these components have on creating the optimal environment for cell survival, growth, and differentiation. They influence transduction efficiency, expansion yields, product consistency, and even final product potency. So if we look at the general process for CAR-T therapy, the cells, once extracted from the donor, will go through an isolation process of the mononuclear cells, followed by cryopreservation until they are thawed again to go through a gene editing step, where the cells are transduced with a virus or transfected by electroporation. These steps are very critical, and their success is closely related to how much the cell microenvironment helps in maintaining cell integrity. This also applies to the process used for stem cell therapy, where the cells, since isolated, need to be maintained in a solution that preserves cell functionality. The cells are also cryopreserved, which highlights the need for an effective cryomedia. These processes highlight how cell culture media and buffer are deeply embedded in the core process of cell therapy. So cell culture media, being the core element for successful therapies, might actually hold your progress back if not properly used. So the lack of transparency is the main culprit, as proprietary formulations are often black boxes. You don't know the exact composition, concentration, or ratio of components. And this makes it nearly impossible to troubleshoot issues, optimize for your specific cell line, or understand why certain batches perform differently. And it also makes it hard to innovate with your therapy and to offer to your cells the optimal condition they need to thrive. So using proprietary media might seem to be the answer for a faster development, but it limits your ability to innovate and optimize your bioprocess. From a process standpoint, solutions transparency and packaging flexibility are crucial for process success and reproducibility. So what really needs to be transparent are media composition, QC data, raw material traceability, and media performance. And the reason is that cell culture transparency provides numerous critical benefits across the entire bioprocessing workflow. So at a scientific and technical standpoint, it enables accurate replication of experiments across labs, helps identify root causes during troubleshooting, supports standardization of protocols across research teams. It helps identify incompatible components interaction, especially when the media requires extra supplementation with the proteins. It helps with process development and optimization. It's also important for regulatory and compliance standpoint, as it facilitates regulatory approval, as FDA, EMA, or other agencies require detailed documentation, supports IND BLA submissions with complete traceability, and demonstrates control over critical process parameters. So this is extremely also beneficial for risk management, as transparency help identifies potential contaminants or animal-derived materials, and it enables proactive supply chain risk mitigation. In terms of economic benefits, transparency helps with cost efficiency, as it reduces expensive failed batches due to unknown variables, minimizes redundant testing and validation work, enables dual sourcing strategies with equivalent alternatives. It facilitates vendor qualification and comparison, supports strategic inventory planning, smooths transition from R&D to manufacturing. It also enables outsourcing to CDMOs with confidence and facilitates site-to-site transfer. Another benefit of transparency is in terms of innovation and collaboration. So as it accelerates development, speeds the process development timelines, enables data-driven decisions, supports collaborative research efforts, and it gives as well a competitive advantage as it builds trust with customers and partners and create a more robust process. And finally, another main impact on of transparency is on patient safety and product quality, as it allows offering consistency in therapeutic production, supports patient safety through controlled manufacturing. It tracks components back to the source in case of recalls and enables rapid response to supply chain disruptions. So the other parameter is packaging. So packaging helps, packaging flexibility actually helps and is key to simplify and solidify processes, reduce human error, allows time saving, reduces environmental impact, improves reproducibility, reduces human error, fastens therapies access, and reduces labor and cost. So cell culture media, being deeply involved in the processes related to cell and gene therapy, is important at all these different stages. And here we have a breakdown of the differential impacts of media and buffers at each stage. At the cell collection stage, the media helps maintain viability, and the buffers help stabilize the pH and prevent clotting. During the activation phase, media and buffers help support a healthy cell activation. During transduction or transfection, using the appropriate media and buffer maximize vector uptake and protect the cells and preserve vector infectivity. During the expansion phase, which is a very critical phase, the media and buffers used ensure reproducible growth and prevent metabolic stress. The media also protects cell markers and cell integrity during the harvesting phase, and the buffers allow for a gentle removal of impurities. Also, media and buffers are involved to maintain cell potency after gene editing, and the buffers help stabilize the product ready for delivery. Finally, at the cryopreservation phase, using the appropriate cryomedia prevents ice damaging, and the buffer enhances post-thaw recovery. It might seem easier to use off-the-shelf proprietary media, but that ease comes at the cost of an increased risk. Given that all media composition available for cell therapy are blinded and their specific formulation is not shared to support you, it actually adds another layer of difficulty to your development. But prior to the era of cell and gene therapy, cell culture practices for research purposes were mostly relying on this closed formula of the basal media that is always supplemented with animal or human protein sources. We all know that this media composition is widely different and wanted to plot it in one graph, as you can see here on this slide, for a better comparability. So representing this set of data that shows that there are components commonly present in all these base formulations. We find usually in basal media four subgroups consisting of vitamins, amino acid, inorganic salts, and carbohydrates. Things we see very commonly in all media are things like choline chloride, folic acid, myoinositol, niacinamides on the vitamin portion. On the amino acid portion, the most commonly present amino acids are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine, which are the ones that we find commonly in these media. The inorganic salts are mostly added to ensure osmotic balance, and the glucose is most commonly used as a carbohydrate source. Other components are very unique to certain basal media, such as galactose, lysine, cysteine, arginine, asparagine, and these specific elements are the one making some cells thrive in one media and not in the other. So these heat maps, as simple as they look, show that different cells have different requirements at a single component level. And interestingly, during the transition towards developing serum-free media for cell and gene therapy, these basal media were used as the base or the starter, supplemented with either blood-derived or recombinant proteins. When we plot the data to show quantitative evaluation of these media, we see a very big range in terms of the quantity of amino acids, vitamins, salts, and sugar present in each one of these media. When we link each media usage to the most commonly used cell type, we can see that cells such as MSCs mostly thrive in different bases, such as RPMI, DMEM, IMDM, and HAMCEP12, as long as these media are supplemented with FBS. However, other cell types, such as HEK cells, mostly thrive in one or two of these media, like L15 and MEM, showing less tolerance to thrive in any type of media. Blood-derived cells such as T cells and NK cells are mostly grown in a base of RPMI, DMEM, or IMDM supplemented with protein source. So to be able to successfully drive your cell culture practices, consideration needs to be given to the components selected and also to the quantity required for each of these cell types. We know that different cells have different needs, but what if they were sharing the same microenvironment in the body? Do we expect, for example, T cells and B cells to thrive in the same media? If we look at this example from a study published in 2019 comparing the metabolic activity in terms of leucine uptake, oxygen consumption, and extracellular acidification, significant differences are noticed. Indeed, I found this example very compelling as leucine uptake, which is involved in protein synthesis, glucose, and lipid metabolism, is lower in T cells compared to B cells. And through their differentiation steps, B cells show very low OCR and ECAR compared to T cells. This translates in different metabolic needs for these cells to selectively expand and differentiate. Indeed, low ECR and ECAR mostly correlate with a lower number of mitochondria in the cells and their activity. So the more mitochondria count is high, the more the cells will rely on OXPHOS over glycolysis and beta oxidation. So these metabolic differences between the two lineages are established during early lineage differentiation and were functionally significant. So the results show here that B cells have higher level of protein synthesis, while T cells have a combination of higher energy demand and lower protein synthesis. In this example here, this was an investigation to identify the influence of 5 cell culture media conditions on T cell metabolism, proliferation, differentiation, activation, and function. So here they refer to IMXV media as REP, immunoccult as ICM, TextMax as TAC, Corning media as CMM, and HPLM. Although each condition supported cell proliferation, the extent of expansion varied, as did differentiation, function, mitochondrial phenotype, and metabolism. Many of these phenotypes were dictated by the media at the time of activation rather than the precise method used to activate the T cells. So this study highlights the effect of cell culture media on the metabolic program, phenotype, and function of human T cells. So these data further highlights the notion that the choice of appropriate cell culture media requires full consideration. So to further confirm these observations, we supplemented biotin and riboflavin back to the depleted NB-ROC and compared its performance to the depleted one. And the data we generated showed a correction of the gap that was marked due to the lack of these 2 vitamins and led to a significant increase in absolute GFP cell count. On another study, we compared the gene expression in T cells from 3 different donors when the cells were activated and expanded in 2 different cell culture media, either in NB-ROC supplemented with 2% physiologics or in optimizer supplemented with 5% human serum. So transcriptomic data revealed differences at the mRNA expression level when the cells are grown in different cell culture media. Interestingly, most of the significant differences were marked in genes involved in cell organization, protein expression, and immune response level. So this study shows that the media used does have an impact, actually an in-depth impact on the cells it's growing, even at the mRNA expression level. So off-the-shelf media looks appealing at first glance. It's convenient. It's readily available. You can order it today and have it tomorrow. But let's talk more about what you're actually getting, or more importantly, what you're giving up. With off-the-shelf comes limited transparency, difficult optimization, higher long-term cost and supply chain risk, while optimized media comes with strategic advantages by offering full visibility into components, optimized performance, cost control, supplier flexibility, and more. So, when designing your therapy, media matters as much as the cells, the genetic modification, and the bioreactor. They are inseparable components of a single system. So, because we support the idea of customization, we wanted to make custom formulation development process move faster than starting from scratch, which led us to the creation of QuickStart, which is our platform for starter formulations. So the idea is that you would start with a pre-optimized media developed in R&D, then going through our formulation service. We further customize each formulation to your need. And after selecting a successful formulation, we end up having a custom media to transfer to manufacturing for scale-up. This formulation will be completely transparent in terms of material sourcing, component, and also in terms of gram per liter. QuickStart Media represents our xeno-free and serum-free pre-optimized media platform. It's designed to give you the best of both worlds, the speed of pre-formulated solutions with the flexibility and transparency of customization. So in this family, we have NB-KUL DF, which is our DMSO-free cryomedia. We also have NB-KUL 10, which contains 10% DMSO. We have NB-ROC as well, that is a T-cell specialized media to grow T-cells and expand them. We have HEKima Adherent, which is developed specifically to grow HEK293 cells in addition, and NB-MSCs specifically developed to grow mesenchymal stem cells. And coming soon, as addition to this platform, we will have HEKima suspension, specifically designed to grow HEK293 in suspension, and as well as NK cell medium. So these formulations serve as validated foundation that you can customize based on 3 key drivers. Performance gaps, as QuickStart Media gives you a proven baseline to modify and optimize until you hit your target. Component exclusion, with complete formula transparency, you can identify and exclude components while maintaining media functionality. And also protocol and process-specific customization. As your protocols have specific requirements, QuickStart Media can be tailored to match your exact workflow, equipment, and timing. This will lead to the following benefits for our customers: faster development and customization, complete formula transparency, full traceability, supply chain security, optimized CQA performance, and the support from cell culture experts. So now let's talk about the service features. Our turnaround time is between 3 to 5 months for formulation services. QuickStart Media actually dramatically compresses your development timeline by giving you that proven starting point. So the customer owns the final successful formulation. This is critical. You're not developing proprietary media that we license to you. When we arrive at your optimized formulation, it's yours. You own it, you control it, you can take it anywhere. And this isn't a vendor relationship, it's a partnership. So multiple rounds are possible with different number of formulations to test, 5, 10, or 15 formulations per round. We can go as low as 500 ml per formulation. And if you need to adjust pH, to change osmolarity, modify growth factors concentrations, exclude specific components, add proprietary elements, all of this is on the table. The platform is designed for adaptability and not rigidity. So sometimes you don't need to reformulate everything. You just need to dial in one variable. Maybe it's glucose concentration. Maybe it's a specific amino acid. Maybe it's the buffer system. QuickStart Media allows targeted single component optimization, which means faster and more cost-effective iteration. So the QuickStart Media platform isn't just for research and development. These formulations are manufactured to be GMP compliant after complete customization. When you're ready to transition from development to clinical manufacturing, you're not starting over. You're scaling up a formulation that already had been produced under the appropriate regulatory framework. So let's start with NB-KUL DF DMSO-free, which is our chemically defined serum-free and zero-free cryopreservation media. NB-KUL DF effectiveness was tested on multiple cells, and here I'm presenting first the PBMC. So it was tested on peripheral blood mononuclear cells. This set of data shows equivalent performance of NB-KUL DF reflected by equivalent viability, cell count, expansion after thaw compared to DMSO-containing formulation. And more interestingly, we see a better performance from an NB-KUL DF compared to DMSO-free counterpart. T cells were isolated from PBMCs and were cryopreserved in either NB-KUL DF, CS5, or CryoStore CSB. As you can see on this graph, NB-KUL DF showed equivalent performance to CryoStore CS5 and better performance compared to CSB in terms of cell viability and cell count after thaw on multiple donors. And to further evaluate its performance, NB-KUL DF was used and tested on MSCs, mesenchymal stem cells. And as the data shows here, NB-KUL DF showed a comparable performance to CryoStore CS5 containing 5% DMSO, or better performance compared to CSB in terms of cell count, viability, and expansion after thaw. So when tested on PBMC's isolated NK cells, NB-KUL DF performance showed slightly lower compared to DMSO-containing media in terms of cell count and viability on 3 donors after thaw. But then when we compared to the expansion, we see that there's equivalent expansion between NB-KUL DF and CryoStore CS5. However, the counterpart, DMSO-free media CSB, showed lower performance compared to NB-KUL DF. And this data is a good example to show that the media needs to be specifically developed for certain cell types to accommodate the specific needs, and that in this situation, media optimization is the answer to improve NB-KUL DF performance on NK cells. So second to the family, we have NB-KUL-10, which is our DMSO-containing media. It is serum-free, xeno-free, and it's used on different cells, as I will show in the following slides. So this slide shows the results obtained from testing NB-KUL on mesenchymal stem cells for cell count, viability, and proliferation after thaw compared to its counterpart CS10. And as you can see, our data shows equivalent performance between the two tested cryomedia. So interestingly, when tested on Adherent cells, NB-KUL-10 showed significantly higher performance compared to CS10 in terms of cell count and viability after thaw. And once the cells are plated for expansion, they showed comparable proliferation. NB-KUL performance was also tested on T cells isolated from PBMCs across 3 donors, and we were able to see equivalent readouts in terms of cell count, viability, and T cell expansion after thaw. And finally, when we tested on NK cells, we also found comparable results when we compared to CS10, and we obtained equivalent performance for both Cryomedia in terms of cell count, viability, and cell expansion as well. Third to the family, we have NB-ROC. As I presented early, it's a media that was specifically designed to grow T cells. So our internal data supported by other labs showed that using NB-ROC for T cell expansion leads to outstanding results compared to optimizers supplemented with either human serum or physiologics in enhancing over 4 times more T cell transduction compared to off-the-shelf counterparts across 3 donors. And in a head-to-head comparison, NB-ROC outperformed industry standard media in terms of absolute GFP-positive cell count, which is a parameter that links between cell proliferation and transduction efficiency, which means that in your process, you will end up having more efficacious cells. And then we also have HEKima Adherent in our platform, which is a media that was specifically developed to support HEK293 cells growing in adhesion. And HEKima performance in terms of cell expansion was tested as well compared to 2 competitors, Sponsoring 293A and Pro 293, and the data shows superior proliferation when the cells are expanded in HEKima Adherent. and to compare protein expression on cells grown in HEKima versus DMEM plus 10% FPS, a lentivirus expressing GFP, was used to transduce the cells. The data obtained showed a very significant increase in GFP expression when the cells are grown in HEKima Adherent compared to the control. So whether you're doing protein production or viral packaging, this media can deliver improved yields and consistency without the variability of serum. HEKima Adherent has been tested across multiple HEK variants, and the data obtained is further confirming the need for customization. While it performed at equivalent levels to DMEM plus 10% FPS for HEK293 and HEK293FT, it showed lower performance with HEK293T, which means having access to formulation developed for a specific cell type does not mean it will show the same performance across different variants of the same cell line. And then last to the family, we have NB-MSC, which is our media developed specifically to grow mesenchymal stem cells. The data we generated shows that NB-MSC supports robust MSC proliferation compared to basal media supplemented with either 5% FPS or 10% FPS. And as presented on this graph, we see a significant improvement on proliferation kinetics and cell confluency when the cells are grown in NB-MSC, proving a defined, scalable solution for stem cell culture and expansion. And then we also wanted to evaluate the stemness of cells comparing cell growth in NB-MSC versus DMEM 10% FPS. And as you can see here, we ended up having equivalent CD90 expression in both cultures. So multiple customers showed interest in our formulation service and QuickStart platform. So we had customers specialized in stem cell therapy. They wanted to develop a serum-free media for MSC. So we used our MSC media platform, which is NB-MSC that I just presented as a starting point. And the result was that they had a unique formulation that they own and that fits their protocol and application with an optimized cell feeding schedule. The second case study is about a customer working on developing devices for research purposes, and they had a formulation they developed internally, and they wanted our support to optimize further the formulation and help them scale up. So customization was successful, and they now have a formula that they own and that has equivalent performance to proprietary control buffers. And the third example I'm showing here is about a customer working on developing a CAR-T therapy device that requires the cells to grow under specific conditions. So we used our T-cell media as a starting point, which is NB-ROC, and now they select the best version of NB-ROC that performs well under their protocol and that meets their CQAs. So no matter where you are in your media development, we will meet you there. So we start with production volumes that match your early development needs, up to 50 liters of RUO media. Whether you need 10 liters, 1 liter bottles, or smaller formats like 1 liter, 5 liter, 10 liter, or 20 liter bags, we provide the flexibility to support your research and early stage work. We dial in pH and osmolality to match your requirements, and the key here is the quick turnaround. We know speed is critical in early development, and we deliver accordingly, usually within 2 to 3 weeks. And using our formulation service is where the real customization happens, and we offer different pathways depending on where you are in your journey. Using Nucleus Biologics expertise, we develop media buffers based on your critical quality attributes. So the first path is when our customers test the media performance and share feedback with us in an iterative process. Typically, this takes 3 to 5 rounds over about 3 to 5 months to reach a final optimized formulation depending on how fast the customer can generate the data. But on our side, we typically develop and ship media within 2 to 3 weeks for each round after receiving the performance feedback. The second path is if you need wet lab support to evaluate media performance, as we can develop and test your media performance after protocol transfer or on a proxy cell line. But maybe you already have a head start. If you developed your own media and want support for gradual scale-up, we also offer media production and wet lab support to consolidate data on your developed media. We provide the testing infrastructure to validate and refine what you've already created. And our QuickStart Media platform is our accelerator for formulation services development. So QuickStart Media is key to speeding up the media development process. It gives you a validated starting point that dramatically shortens development timelines and gets you to an optimized formulation faster. So all these paths lead to the same destination, which is an optimized formulation tailored to your process. So once your formulation is locked, we handle the tech transfer and scale up all the way to 2,000 liters. We manage every detail from raw material selection and qualification, process development, filter studies, package qualification. We establish method suitability and qualification for USP methods. We conduct as well QC and release testing with proper method transfer. We also handle shipping validation to ensure your media arrives in perfect condition. We also provide complete GMP documentation with strict specification design, batch record development, quality and regulatory support for submissions, commercial scale-up GMP manufacturing capabilities, and shelf life studies to support your regulatory filings. So we don't just manufacture media, we partner with you to develop, optimize, and validate and scale formulations that are designed for your cells, your process, and your regulatory pathway. Because when you have complete transparency and control over your media formulation, you're not just following best practices, you're actually defining them. So we've talked about the science of media optimization, the strategy of customization, and the platform that makes it possible. Now let me show you the manufacturing infrastructure that brings it all together. Because having the right formulation is only half the equation. You also need the manufacturing capability, the quality system, and the operational excellence to deliver that formulation consistently, at scale, and on time. So at Nucleus Biologics, we've built what we believe is best-in-class rapid turn manufacturing. We've streamlined our processes to deliver GMP-grade buffers in 4 weeks without compromising quality or documentation. We have 11 dedicated ISO 7 clean rooms, which means we can handle multiple projects simultaneously, segregate incompatible processes, and maintain throughput even during peak demand periods. Our segregated dispensing suites ensure that your media and buffers are prepared in dedicated space, eliminating the risk of product carryover from other customers' formulation. We also operate in liquid clean rooms equipped with ISO 5 BSc. So this is the gold standard for aseptic processing, providing the particle control and air quality needed. And whether you're in early development with small research volumes or scaling to commercial manufacturing with multi-thousand liter, we have the equipment and processes to support you. We offer complete flexibility in final container formats, whatever works best for you and for your downstream operation, whether small vials, bags for bioprocessing, bottles, or drums for bulk application. And our aseptic processes are formally validated using media fills and other industry standard approaches, which gives you the confidence and gives regulators the documentation they expect. We conduct as well active environmental monitoring throughout manufacturing operations, viable and non-viable particle counts, surface monitoring, and personal monitoring. Our water for injection, Wi-Fi water, are sanitized using ozone, providing consistent, high-purity water that meets USP standards and supports your formulation integrity. And finally, we have our batch record system, MediaVerse, which is our electronic batch record system that ensures complete traceability, real-time data capture, and audit-ready documentation for every batch we manufacture. So Nucleus Biologics is one of only 2 media companies in the world with EXCiPACT certification. So this is a rigorous, internationally recognized standard for pharmaceutical excipient manufacturers, and achieving it demonstrates our commitment to operating at the highest level of quality, safety, and regulatory compliance. So having EXCiPACT part of the game helps ensuring product safety and purity, helps with regulatory compliance and GMP standards, helps maintaining consistency and reproducibility, and also supports scalability for commercialization. So let me bring everything together by showing you the complete journey we support, from your earliest research through commercial manufacturing. So working with Nucleus means one formulation platform and one seamless transition from bench to bedside. As we offer our services from early RUO grade development supported by formulation services and our QuickStart platform, we offer formula transparency for a better outcome for your process. We support you during scale up as the requirements become more stringent as you move forward with your therapy by offering GMP and excipient grade services. So, from QuickStart media development through commercial scale production, we have the infrastructure, the systems, and the expertise to support your journey. So, thank you for your attention today, and let us know if you have any question.
All right. And with that, thanks so much, Osma. This was awesome. A lot of great information, and audience members, we are going to get to the Q&A here in just a second. And so if you do have questions, please submit them through that Q&A box. Just so you know, that Q&A box is private. It's just between us and you to ask questions. And so we'll keep that information and your name and everything private. So please feel free to submit them. It's not going to be anything for the public. With that, just a couple reminders, everything you're looking at on your screen. If you did want to reach out directly to Osma as well to ask a question, you should see on the left side of your screen, there's a little speaker bio panel. Click on the little blue circle right below her picture, and then that'll let you message her directly as well if you wanted to ask something that way as an option as well. And then my information is there as well if you wanted to connect with me, if I could help you with anything too. And so with that, let's get into some questions. And so Asma, this first one. So at what point, let's see. So at what point in the development of my media and buffers should I evaluate the benefits of a custom formulation? And then they followed up a second one. Are you positioned? Can you support scale up?
Yes. So, again, the benefits of transparency, knowing what's in your formulation, really helps with further development, helps with troubleshooting, helps with supply chain. Yeah, and as I mentioned early, we do help with scale up all the way to 2,000 liter, and because we have the infrastructure in place, we know how to do that, and we do that with a lot of our customers.
Okay, perfect. I've got another one here. So, are the quick start formulations transparent? And if not, what is the process to customizing starting with those formulations?
So the quick start right now, right away, are not transparent. So to get to the transparency level, we need to go through formulation service where we optimize the formulation to your needs. So this way, the formulation is tailored exactly to the protocol you're using. All the components that you want in or want out of your formulation are taken into consideration. And then after the customization process, then the final formulation that works the best for you, because as I mentioned, we run through multiple rounds with different formulations. So the final formulation, that one, you will have transparency, and then you can own it.
Okay, perfect. Thanks so much. All right, our next one. So, with all your custom media formulations, will they be managed through your QuickStart Media program now, or can I still develop a media from scratch with you?
So it really depends if the cells you're using do fit in our QuickStart platform. Then yes, we start with QuickStart. If not, we use our expertise because QuickStart does not reflect all the expertise we have, and then all the cells that we've been using so far. We can use our expertise to develop your formulation, and then we started from scratch. So we're not only limited to those cell types for the formulation service.
Okay, perfect. So with your GMP services, is every media excipient quality, and can I choose the services I want performed, or is it a package?
So, no, it's not a package. So you can they can choose between RUO, GMP, or excipient. So the way I would identify excipient, you can even call GMP plus, because it adds like with aseptic for qualified, ID testing via FTIR or RAMAN, raw material risk assessment, and like it adds a lot of other layers to the GMP, but it's more stringent and more strict than GMP. But again, like you can pick and choose from the services you're interested in.
Okay, awesome. Let's maybe do one or two more here. And so when assessing media performance for my process, what factors should drive my decision to move from an off the shelf to a custom formulation?
So again, it's transparency. Imagine if you start your development with basically a black box. You don't know what you're using, and then you run into issues with supply chain, with troubleshooting. Sometimes, like you check every single level in your process, and you don't see anything that would lead to any of the problems that you're seeing, and then you're like, okay, maybe I need to troubleshoot the media, and that's where all the issue starts, and that's where the all the struggle starts actually. So having transparency, you can add that to your troubleshooting steps, and then identify at a single component level what needs to be um, evaluated, and then it helps you understand better your system and even improve it.
Okay, awesome. Maybe one more here. So do I have to buy the media from Nucleus, or can I purchase elsewhere?
So, because we give you the transparency on the media composition, for sure you can take it elsewhere, but there will be a supply agreement for a while. Like those are like few details that could be discussed, but again, like as you have the formulation, this is something it gives you the freedom to take your formulation elsewhere as well.
Perfect. All right. Awesome. Thanks so much. And I think with that, this would be a great place to wrap up. Thank you, audience members, so much for joining us today. If you had any more questions, you can still submit them through the Q&A, and I'll pass those along to everyone here for some potential follow-up after the event. And then, Asma, again, thanks so much for joining me today. This was great. I appreciate your time. And audience members, thank you so much as well. We appreciate you being here. If you need anything else from us, please reach out directly, and have a great rest of your day. And we'll see you next time. Bye, everyone.
Thank you. Bye.